So precision or personalized cancer medicine really involves taking care of all the information available about a particular patient and his or her cancer and drawing in the world's experience in order to design therapy that will be successful in either curing the disease or prolonging their life. And because we're able to measure mutations in DNA effectively today, and because many of the new drugs available target the products of these genes, most of the emphasis in personalized cancer medicine today is on gene sequencing of the tumor to find out what's driving that tumor and then giving drugs that will affect the tumor. So the paradigm here is we now know most of the genetic aberrations that can cause cancer. The list is roughly 200. There's over 800 drugs in the pipeline. A few are approved, many more are experimental, that target the products of these abnormal genes. We can detect them, as I pointed out, and they can become biomarkers that can pinpoint an individual patient where we have evidence that they're more likely to respond to the therapy because the gene abnormality the therapy was designed to block is present in their cancer. And we can do that in a reasonable time frame now and at a very reasonable cost. It's about the same cost as 2 MRI scans, which are routinely done on patients. And clinical trials using this approach have been successful. So the first clinical trial that tested this hypothesis was designed by Ki Hong, my colleague at MD Anderson, and it was published in 2012, but he wrote the grant and planned it beginning around 2005. It was funded by the Department of Defense, not by the Cancer Institute. DARPA is a fabulous resource in this country. It founded the internet. It's willing to take chances, and in this case, they funded this trial. So this is a cartoon of a cell, and you can see there are 4 yellow molecules, which are the products of genes that we know can cause or promote cancer. The fourth one is in the blood vessels over there. VEGF is not a cancer gene, but it promotes the growth of the blood supply to the cancer. It's called angiogenesis, and I'm sure you're all aware of that. And there were experimental drugs available that were designed to attack the products of the genes, these 4 genes that are in yellow. And the question Dr. Hong and his colleagues asked, in routine phase 1 trials, where you're just beginning to study a new drug, the drug is given to patients with advanced cancer, and you look at the side effects, and you look at the efficacy, and the response rate is usually about 8%. Most of the time, the patients don't respond to those drugs. But Dr. Hong and his colleagues postulated that if the new experimental drugs were assigned specifically to patients that had the genetic abnormality for which that drug was designed, those patients would have a better response rate. And this trial was carried out. It required biopsies of people with advanced cancer, which is of the lung, which there was a lot of argument at MD Anderson of whether this was ethical to require a patient who had advanced lung cancer, had failed all forms of therapy, should go through a biopsy, which can have side effects. And they already have deteriorated lung function. Finally, after about a year or so, it was agreed that we could do the experiment. It turns out that about 250 patients were interviewed, and only 1 patient turned it down because of the possible complications of the biopsy, which of course those are explained in the consent form. And all the rest of the patients said, please give me the drug, I'll do anything. I'm dying of cancer and I hope it works. And of course in phase 1 trials, it's rare to it's almost never the fact that we can cure cancer, but we can prolong life. And this study showed that if you gave a patient a new experimental drug that was targeted, they did better than if you just randomly tried experimental drugs. And that was a changing paradigm in how clinical research is done. And the other study done at MD Anderson was really done by our Department of Investigational Therapeutics, doing a variety of trials with a variety of different drugs over a period of around 3 or 4 years. And they collected data on 175 patients on the left there, where the therapy that they received matched retrospectively, looking at their tumor. They didn't have sequence data on the tumor at the time, but it turned out that in 175 cases, they got an experimental drug that targeted a gene that was aberrant in their tumor. And they compared that to 116 patients who got an experimental drug where it was irrelevant what was going on in their tumor. And it was a highly significant difference. The complete response rate and partial response rate was 27% in the matched group and only 5% in where they just randomly got the experimental therapy without any correlation with what was wrong in their tumor. And the diagrams, in those diagrams, red is bad and green is good. And you can see the ones that got the match therapy. Many more patients had their tumor shrink, shown in the green part of the graph, compared with the controls, and stabilization of disease for more than 6 months, which was a big deal in people with advanced lung cancer at that time, with an expected lifespan of 3 to 4 months at the most. Was 23% compared to 10%. So again, evidence that an approach to the ethical question, is it fair to tell a patient, this is the way you should be screened and treated when you're put on an experimental drug? The answer was yes, this was quite ethical, in fact, favorable for the patient. Now, this is a summary of 6 different experiments that were published that worked. There are others that didn't work, and many of them aren't published, and so we don't have a denominator here. But in each of these cases, the experiment with the patient, the clinical trial, would have failed unless the patients had been pre-screened, and only patients were put on the trial who had the gene abnormality for which that drug was designed. So Herceptin, which is one of the great drugs against breast cancer, the trial that led to its approval by the FDA, required that a patient have high levels of Herceptin in order to get on the trial. And the trial was positive. And if that requirement had not been there, the trial would have failed. Less than 10% of patients would have responded, and the drug would have been shelved. Imatinib, which is a very good drug against chronic myelocytic leukemia, you don't need to pre-screen there. It turns out that stomach tumors that have a c-KIT mutation also respond to imatinib. They're not common, but the experiment was done testing whether imatinib worked against stomach cancer. If it hadn't been pre-screened in those patients, the trial would have been negative. But it did work. It didn't cure the patients, but it prolonged life substantially, years. And the other 4 are examples, and I'll just pull out number 5, crizotinib in lung cancer. Crizotinib was developed to treat lymphoma. It's a disease of the cancer of the lymphocytes with a common ALK, A-L-K, abnormality. About 3% of lung cancer patients have an abnormality of the ALK gene. It's rearranged with the EML gene. If you ran a clinical trial with lung cancer with an ALK inhibitor, crizotinib, the response rate would be less than 3% because only 3% of the patients even have the abnormality. But investigators at a number of cancer centers got together and screened a few thousand patients to find 70 patients that had that rearrangement. They ran the trial. The drug company provided the drug and was enthusiastic, the pharmaceutical company, and over half the patients responded, and the responses, again, in advanced lung cancer, were an average of 9 months and some longer. So this drug was approved, and it wouldn't have been approved without the biomarker. So the take-home message here is that treatment now today with these new drugs is much more advantageous for the patient and for getting the drug to be approved, getting positive results in general if you pre-screen with biomarkers. So with that as background, we set up the Sheikh Khalifa Institute for Personalized Cancer Therapy at MD Anderson, and I was fortunate enough to be appointed the first director of that institute. and I began that as soon as I stepped down as the president of MD Anderson. After taking a 6-month sabbatical at the Broad Institute at MIT and Harvard in order to get up to date on the genomic sequencing research scene, where I think they were pioneers and leaders in that. And our goals were to do genetic analysis on large numbers of patients. If standard therapy, surgery, radiation, and chemo is not producing cure or long-term control of the disease, and they need experimental therapy, we wanted to be able to screen their genes, and we estimate this could be 30,000 a year. We wanted to develop the clinical trials that would bring therapy to these patients. We wanted to provide decision support, advice for the clinicians and their patients, because this is very complicated. The average clinician today does not know a lot about genomic abnormalities in cancer. The average cancer physician doesn't know a lot about it. So there's a whole education program required to bring our treating physicians, except for maybe about 10% of them that are in this already, up to date on this new development. We wanted to demonstrate the value of this approach because in most cases this had to be paid for by philanthropy or by hospital margins because insurance companies do not reimburse experimental therapies. And we wanted to educate the next generation of clinical investigators. Now this slide just shows the various sequencing tests that were used at MD Anderson over a period of about 6 years. And it began with Sanger sequencing, for which he won the Nobel Prize, which was the original way to sequence DNA. And it's working in the future. This year, we're going to introduce Oncomine. Most of the work I'm going to tell you about was done with the ion torrent method, where we're screening 46 genes. We're looking for mutations in 739, so you can do the division, and we're looking at a fair number of spots in each of those genes where we know mutations occur for those 46 genes and then 50 genes. And this is the list of those genes. And when we began this work, we had clinical trials that tested therapies against about a dozen of them. And you can see that by 2014, we had available at MD Anderson clinical trials that tested drugs against the majority of these genetic aberrations in cancer patients. So we were able to, and all of these come from pharmaceutical companies with whom we work, and we convinced them that we would collect the information they would need to advance their products, either yes or no. And the protocol we designed was called Clearinghouse, and the patients, again, the ones who had failed standard of care, because the patient didn't fail, we failed to have a therapy that would control or cure their disease. They were studied, and if an actionable aberration was determined, actionable means, number one, that genetic abnormality is known to drive their cancer, and number two, there's a drug that is designed to act against that particular target. The patient would be put on the appropriate therapy, which is, I never should have pushed that button. There we are. I'm not going to touch it again, but it's the middle arrow going down. And we collected information. And if nothing turned up on the screen, we took the specimen that was left over from the clinical laboratory, which is a highly regulated laboratory, and it's very expensive to run tests in it, and sent it over to the research laboratory and screened 200 genes instead of 50. And if we found something that might be interesting, we had to send the information back to the clinical laboratory because it had to be verified in the clinically careful, with proper controlled environment, so that the hospital wouldn't be closed, and then the patient might get benefit from that. So we've screened about 4,500 patients over the past 3 years. The first 2,000, we started digging deep into our data, and we found 39% of them had potentially actionable mutations in genes in their cancers. And I just defined for you what a simplified version of what actionable means. About 21% had other mutations that were not actionable. Maybe there was no drug for that gene abnormality. Maybe there was an abnormality in a gene that didn't change its function at all or had no known change in its function. 10% had likely germline variants, some of which can cause disease. And we had to bring in genetic counseling at that point because this patient's family had to be made aware of that if the patient agreed. And about 30% had no mutations. And only 11% were placed on genotype matched trials. And the reason that number is 11% and not higher is complicated. We've studied it thoroughly, but these are patients with advanced cancer. To get an experimental drug, they had to come to MD Anderson. The majority of our patients are not from Houston. If you're dying of lung cancer and there's an experimental therapy available and you've got to leave Omaha and come here to live while you're getting your experimental therapy, you sometimes decide, I'm just going to go back to Omaha. And other situations occurred where we had experimental therapies available targeting a gene, but the company was only targeting a cancer where that gene was commonly abnormal. So maybe in colon cancer, this gene was commonly abnormal. Suppose you got a patient with brain cancer that had that gene abnormality. There's no experiment. The company will not let you take that drug they're studying in colon cancer and try it out on a brain cancer patient. They're worried that some horrible side effect will turn up. Anything that will slow down proving that that drug works in colon cancer is detrimental. That's the way they look at it. So sometimes patients are very frustrated because all our patients get on the web and they find out all the trials going on, and we tell them the trials going on. And they say, why can't I get that drug? And you do the mother test. You call the drug companies. If your mother had this cancer, wouldn't you want her to have this drug, even though it's not part of your trial? And they're tough. They say no. That may be changing. Now, here's some of our results in those first 2,000 patients. And across the bottom are listed the genes that we tested. And the y-axis is the patients that had somatic mutations. And you can see that after about 5 or 6 genes, we're at the 1% or less level. So the gene abnormalities, except for a few on the left, which are very important, P53, there's no drugs for, PIK3CA, KRAS, BRAF, IDH1, those are more common, and there are patients that can benefit from these drugs. But as you get to the right side, you can see that a small fraction of patients are found to have an aberrant gene in these cases. And it varies with the kind of cancer. So on the left side, if you have pancreatic cancer, 78.9% of patients had an actionable gene abnormality. On the right, if you had renal cancer, only 5% of patients. Now, this kind of information is very important for people planning clinical research in academic institutions, in practice, and in companies. And the variation is pretty amazing, actually. And there's a very common question in cancer. Well, the patient has metastases, and you've got a piece of the original tumor that shows an abnormality, but how do we know there aren't other abnormalities in the metastasis? And we had here a number, a little over 100 out of those 2,000, where we had biopsies not only of the primary tumor, but of the metastasis. So on the horizontal axis are some genes, and on the vertical axis are some kinds of cancer. And the blue squares are situations where the metastasis and the cancer had the same driver mutation, the same actionable gene. In the 2 green squares, the primary tumor had it, but the metastasis didn't. And there were no examples, there are no orange squares where the metastasis had a gene abnormality that wasn't present in the primary, which is reassuring. It means you can go back to the original tumor and you can pick up at least, for that list of there, about 12 or 15 genes, you can pick up what might be appropriate target for therapy. And these are data from Dr. Mills, who ran the T200 test, which is checking 200 genes. And we're asking the question, if you lump together all kinds of cancer, what percent of them have an abnormality? Now, the BRCA1 abnormality is present in about 10% of cancers. It was first described in breast cancer, but it's obviously present in many other kinds of cancer, and you can see them all listed there. mTOR, down in the middle at the bottom, is only present in about 2.5% of cancers. So if you're a company wanting to study mTOR, you might want to come to a place like MD Anderson where we see a large number of cancer patients. We're treating over 100,000 cancer patients in any one year. Whereas if you are studying a BRCA1 abnormality, it's more common, and you might not need to go to a large institution to find a variety of patients to study. And I want to emphasize, if you take let's say mTOR, and you can see skin is, the blue is the most common, and then breast, the red is the next most common. The fact that a drug works in one cancer against a mutation in a gene doesn't mean it'll work in another cancer that has the same mutation in that same gene, because other things in the environment of that tumor may mean that that gene is not a driver of that cancer, even though it was a driver of the former cancer. There may be bypass pathways in the second cancer, which or other explanations of why the drug might not work there. So we have to test these drugs in every kind of cancer against the gene aberrations that we detect. Our second protocol, which is much more interesting really, is looking at unusual responders to try to understand and learn new information that will help us. So sometimes there are exceptional responders where there's a complete control of the disease. And we'd like to go deep into that and find out all the gene abnormalities, not just those 50, to see why that patient might have responded and the other didn't. Sometimes patient have a response, and then unfortunately, typically, the tumor breaks away and starts growing again. We'd like to rebiopsy that tumor and say what happened to the genes in that tumor, so now the same drug that worked for a while is no longer working. And we're doing that in a variety of examples. Sometimes patients respond in one part of the body and not another. The tumor that metastasized into the lung lesion shrinks. The tumor also spread into the liver lesion doesn't. There's biologic reasons why the tumor may be growing better in the liver than in the lung. We'd like to understand those, and other examples too. So that is ongoing. And we'd like to learn from our patients in that kind of setting. One example of that is studies with a drug called sorafenib that was given because it targets BRAF, which is a common mutation in melanoma. Well, we had a trial going on, and a patient had an exceptionally excellent response to sorafenib with melanoma, and the tumor really got under control, and it was a prolonged response. We wanted to learn why that patient with this BRAF mutation had done so well. So we didn't know the patient had a BRAF mutation at the time. We just knew this melanoma patient had responded and that BRAF was common. Now this is a CT scan, and the arrow points to the tumor, and on the left and on the right the tumor's disappeared, and the little baseball next to the tumor, that's the diaphragm poking up, the normal diaphragm. So the tumor has disappeared in this patient. Well, we went ahead and sequenced that tumor, and it didn't have a mutation in BRAF, but the patient had responded well. Turned out the patient had a mutation in KIT, it's a gene I mentioned earlier. Well, nobody had studied KIT in melanoma, so we went back and looked at melanoma, and in certain kinds of melanoma, 30% of the patients have a KIT mutation. And the majority of those do respond to this sorafenib drug, not designed to attack KIT, but it happens to attack KIT. It was designed to attack BRAF. It's the kind of thing that makes the research interesting and exciting and moves it forward. And here are just some examples, primarily around EGF receptor research from the literature. This isn't from MD Anderson, where, for instance, gefitinib is a drug against the mutated EGF receptor. But if another receptor called MET is amplified and activates another competing pathway, resistance to gefitinib develops. In the case of cetuximab, our antibody against the EGF receptor in colon cancer, it was found that if KRAS, another gene, is mutated, it doesn't work. So that's a negative screen. If patients have colon cancer, it's standard of care now to get cetuximab, but now it's standard of care to check, and if they have a mutation in KRAS, they won't get it. And that saves them time, money, and side effects, and gives them the opportunity to look at other therapies. Another drug, vemurafenib, that targets BRAF. Beautiful work by René Benards in the Netherlands showed that if the EGF receptor is activated, the cell no longer is hurt by blocking BRAF, and with vemurafenib, and the patient develops resistance. And there's trials going on now giving vemurafenib in combination with an agent that blocks the EGF receptor to see if that will work. And there are other examples listed on this slide. So this is an area of laboratory research that's based on the human patient as the subject, not a mouse, and trying to understand how drugs can work better. And in closing, I just want to mention that in the past 3 or 4 years, immunology has become very important. It's been 100 years, more actually now, since Paul Ehrlich tried to develop the magic bullet against syphilis in this case, and that the immunologists were very excited by this. And finally, in the past few years, we're beginning to understand the immune system well enough to begin to incorporate it into personalized cancer therapy and targeted cancer therapy. And the references here, REV means the reviews. The prognosis of many cancers correlates with the number of cytotoxic lymphocytes and natural killer cells in the tumor when it's biopsied. It's not an absolute predictor, but there's a definite strong correlation. And in colon cancer, if the tumor specimen contains a reduced level of interleukin-15, which is very similar to interleukin-2, it's a stimulating molecule that stimulates lymphocyte function and proliferation. If that is low, the response to therapy, any kind of therapy in that tumor, is worse, and the patient has a poorer prognosis, suggesting that the immune system is important. The drug Herceptin, which we talked about, trastuzumab, which was designed to block the HER2 receptor, it's now been shown that probably it works at least as much by mediating an immune response, because it is an antibody, to the tumor rather than just by blocking the receptor. Probably works both ways, as does our antibody. And Dr. Allison, now at MD Anderson, and a Texan originally too, did the really important work showing that if you block certain receptors on lymphocytes that turn them off, you can unleash an immune response against a substantial fraction of cancer cells and produce long-term remissions where the immune system now is able to fight the tumor because you've taken the brakes off the system. So there are a number of examples now where chemotherapy and targeted cancer therapy interact with the immune system. And one of them is in breast cancer and prostate cancer. The gene called PTEN, if it's lost, and that is a common finding, maybe in 20 or 30% of some cancers, there's an increased production of PD-L1, and PD-L1 is the protein that blocks the lymphocytes from carrying out their immune function. And summarized recently. BRAF we talked about. When it's inhibited, there's transiently an increase in T cell or killer cell infiltrates in melanomas, an increase in antigen expression in the melanomas, and reduced immunosuppression by cytokines like interleukin 6 and interleukin 8. So interleukin 15 stimulates the immune system, interleukin 6 and 8 block it. And there are dozens of molecules that stimulate and block the immune system. It's a very complicated area. But when the patient's treated with BRAF, after about a month, this PD-1 and the PDL, the ligand, PD ligand 1, increase in expression, and that will, of course, turn off an immune response, suggesting that if you're giving a combination of therapy, you might give BRAF, wait a month, and then give a drug that will block the production of PD-1 and PDL-1, and that's being studied right now. The response to neoadjuvant adriamycin or Herceptin, trastuzumab. Neoadjuvant therapy means you're giving the chemotherapy before surgery to shrink the tumor. And it's been shown that is beneficial. The response is predicted by the biopsy of the original tumor if there's increased CD8 cytotoxic T lymphocytes or if there's a decrease in regulatory T cells. That will predict whether chemotherapy with adriamycin will shrink the tumor substantially in a breast cancer patient before the tumor is removed definitively by surgery. Again, interactions between the immune system and the targeted, in this case, adriamycin isn't even a targeted therapy, it's a standard chemotherapy. And there's a lot of evidence that the intratumoral T cell content correlates with more effective elimination of ovarian cancer when the patient is treated with platinum, another chemotherapy agent. So lymphocytes potentiate chemotherapy. And it may be that many cytotoxic chemotherapeutic agents kill cancer cells in certain ways which stimulate an immune response against the cancer cell. So challenges for the future, and my talk will be about an hour. We did get started a little bit late. I'm going to summarize two major challenges. One is we need more trials with combinations of therapies, and from what I've just told you, that will include immune therapies. We need the biomarkers so we can pick the right patients, because otherwise the trials will be negative. It's not good for the patient. It's not good for the drug company. We need to work out the optimal combinations based on these biomarkers. Often we'll have to work with multiple drug companies. They sometimes don't want to work together. They're getting much better at that. We have to get, when we start combining drugs, we have the challenge of combining toxicities. We have to get more able to study drugs in infrequent situations. I gave you the example of the colon cancer drug and the patient with brain cancer who had the same genetic abnormality. I'd like to be able to study that drug in that brain cancer patient, and the patient would certainly like it. And we need to then understand sensitivity and resistance better. But the other area is more our fault and not the cancer's fault. We're collecting huge amounts of data now. Every time you sequence a genome, you're getting 3 billion pieces of information, and we're doing it on thousands of patients over the country and over the world. There's no standardized, interoperable way to collect that data. The data is often balkanized. It's in silos. The companies don't want to release it. The investigators at the university don't want to release it until they publish their 3 papers on it. They don't want somebody else finding out what they spent 2 years studying. These are reasonable reasons to hold on to data, but it's slowing down what we could be learning. We need a vast knowledge network where we share the data. Then the other thing is we need the decision support tools to help oncologists practicing with their patients to use the information. Because, again, in medical school until 10 years ago, this wouldn't have even been discussed. So we have a website, Personalized Cancer Therapy, PCT MD Anderson, with a decision support tool, which we've developed, which allows, in this cartoon, selecting the blue drug for the blue patients and the orange drug for the orange patients. And right now we have information on that website for the 26 most actionable genes that you saw on one of the charts there. And we give a lot of information, including access to the primary publications by clicking. And we discuss the therapeutic implications and the level of evidence for each therapy and the clinical trials that are available. And we have information now on the trials available at MD Anderson, and we'll soon be loading in the information on trials anywhere for the patient. And the other thing I'll just mention is that we've formed a worldwide innovative network. There's a consortium that's trying to advance personalized cancer medicines, and it includes academic medical centers, pharmaceutical companies, biotech companies, non-for-profits, and governmental agencies there are 40 members from 4 continents. We have a symposium every year where everybody unloads information to try to find ways to move things forward more quickly, and we're developing innovative clinical trials, which I'll just summarize very briefly. On the right side, the Winther trial, I told you that only 39% of our patients have actionable genes. Well, that means that 71% of the patients can't get the experimental therapy. But the experimental therapy might work even if the gene isn't mutated, if the gene is overactive. So we're looking at expression levels, RNA, to see which genes are highly activated. And we're testing the hypothesis that even if your gene isn't mutated, if it's highly activated, that may still be a valid target for an experimental therapy. And it's an innovative trial. It's ongoing now. It involves a whole lot of complexities that I won't get into. And we have a series of other trials for stage 1 and 2 and 3 and 4 lung cancer, where we are designing innovative ways to try to take the knowledge we have and study it in patients from China, India, the Middle East, Europe, North America, and South America, which means if there's some genetic variation due to race, due to what you inherited, that we don't even know how to ask about, we can get around that and check the drug in a variety of people. So in the future, I think this is going to become more and more important for every patient, getting a profile of molecular markers and immune markers, which aren't available right now in any useful way. The environment around the tumor is going to be important. Prostate cancer metastasizes to bone because in many cases, bone produces chemicals that promote the growth of the prostate cancer. We don't know how to study that yet, but we might be able to. And the general physiology and the psychology of a patient is individualized. If you're overweight, you don't do as well with breast cancer as if you're trim. This is useful information. We don't know how to use the information right now. You've already got the breast cancer. You've got the overweight patient with you. I don't know whether making that patient lose weight after they've got the cancer is advantageous. In fact, it might not be, because cancer patients lose weight on account of the cancer. We're trying to help them gain weight. But there's a lot of information that we don't know how to use yet. So that's the background of the experiences I've had. And there's time for questions. And again, it's just been a pleasure talking with you and telling you through my set of spectacles the adventure of what's happening in cancer today and in the area of molecular biology and genetics, where they've come together in ways that are very exciting for helping patients and will help more and more patients over the next decades. Thanks very much.